All right. Let's rock and roll, he said in the music classroom. Well. Yep, happy Tuesday afternoon, everybody. Welcome back. It's been a long 3 days break since we were 4 days break since we saw each other last. Hopefully this Tuesday-Thursday schedule doesn't make it more difficult to learn things and remember things about genetics. Brief comment about class structure. So feel free, you can carry on working on your Socrative exercise if you want. Again, normally when we start class, there will be some sort of an entry quiz. Normally that's just for me to take attendance and to give you opportunities to see the sorts of questions I like to write. So this is an opportunity always for you to get feedback about the sorts of questions I might be asking on exams. These quizzes themselves, as I've probably said before, are just a single point a day or a single point, maybe I'll grade every other Socrative quiz that I give, just for attendance and a point a day or a point every other day. So it's small points, but this is one of the main ways that you earn points for this class. The other way that you'll earn a lot of the points is in exams here in class. And that's why being in class is so important. So I'm thankful for all of you that showed up today. So you're earning points. The other way that you earn a lot of the points in class is by conducting those Google Classroom distributed exercises like the two that I asked everybody to try to complete for last night. Again, my philosophy on all of this is that these are opportunities for you to see what sorts of questions I write and like to ask, and for you to have opportunities to get feedback on your understanding. So the goal for both these Socrative quizzes and the Google Classroom distributed exercises is not necessarily for you to first time get the right answer. So I'm scoring Google Classroom assignments and these quizzes not for correctness. I'm just giving you incentive by offering points for you to actually do them and get feedback for yourself on whether or not you think you understand the material. What we spend in class time doing now is talking about the answers to those questions. So this is where I hope you'll ask me questions about what you didn't understand about the Socrative questions you're answering right now, the exercises you tried to complete for last night. So I hope I'm not stressing anybody out by offering all of these opportunities for you to get feedback. They're not all scored. They're for you to get feedback for yourselves. They're not me assessing your knowledge. They're you, hopefully, self-assessing your knowledge. That's my philosophy and my approach. And if you ever have any specific or other questions about grading, what exercises might or might not be graded, please ask. So it looks like people are getting pretty close to done with those Socrative activities. Are there any questions I can answer right now? We've got a couple minutes for class, questions about class, questions about assignments, course structure, concerns. Okay, so I'm going to move on while a few of you are still finishing up. I want to share this with you because I want you to know that you're not alone in the way that you're feeling about this class and about being students. So you might recall I asked through a survey a couple of weeks ago why being in this class and succeeding is important to you. Passing is important, genetics, biology, class. What's the next biggest word? Graduate, probably. Right? So for a lot of you, this is a required class, so you have to take it to graduate. You have to pass it to graduate. A lot of you are biology majors, you have to pass this class to go on and take cell biology. Okay. A lot of you also said it was important for learning because you're going to hopefully apply this material in medical school and other ways after you graduate. So you're all here together to do those things, sure. Yeah, everyone's here to pass. Hopefully I can motivate a few more of you to do more than just pass. More importantly than that, most of you, or a lot of you at least, are here. I was so happy to see this. After getting a degree, what's next on why most of you are motivated to be here and succeed? Family. Making your family proud, supporting your family, doing something for your family. Right? That speaks to me a lot, and I'm glad to see that that's a main motivation for a lot of you to succeed in this class. And I'm here to help you succeed for yourself and for your family. So anytime you have any questions, concerns, please, please come talk to me. I'm open to feedback. That's why when I asked in one of the quizzes previously what some of your fears are about this particular course, genetics, let's see, time, work, technology are some of the main ones that come up, right? Feeling overwhelmed, feeling like you don't know what's going on, you are not alone. My intention is not for these to be barriers to your success. So that's why I'm saying, if you have concerns, questions, please come talk to me about them. The last thing I want to do is throw up barriers to your success, especially regarding technology and the amount of work. I used to be in your seat, and I had those professors which, after I saw this, the first thought I had was, I became that professor that I said I hated when I was an undergraduate, you know, the one that thinks that their class is the only class you have. Tons of work, right? I don't want to be that professor. So give me this sort of feedback, and I will do my best to help you manage the coursework. Okay, help you prioritize what's the most important thing to do. What do you think is the most important thing to do to prepare for this class at this point in your understanding? Yeah, like the previous semester students said, watching the videos before class is important. And then for you to get feedback again, to try to attempt the exercises that I set. Most of them aren't graded. They're for you to get your own feedback and understanding. And then we spend in-class time doing the answers. So it's flipped. Instead of me asking you to spend time outside of class doing homework, questions, end of chapter questions and so forth, where you get really stressed out because you've got to turn this in because it's going to be graded, I want to do that work in class. I don't want to give you the information in class and you spend out of class time worrying about points. The points are going to happen in here. So you have to have the content learning just on your own, try it by yourself outside of class. So I'm hoping it doesn't take extra time outside of class. This is just meant to be more productive for you. Now, everyone's almost done with that Socratic quiz. We'll talk about those answers in a little while. I wanted to make sure I got to this point because I didn't finish this the end of last class. The main question we were talking about last class was, why do chromosomes have stripes? And I never got to answer that question. Why do chromosomes have stripes? And specifically this question of, what does an isochore have to do with chromosome stripes? So isochore, what was it?
Section of the chromosome with a high GC content or a high AT content.
Okay, so here's a part of human chromosome 14 that is higher than average, average being this horizontal line there. So there's a part of human chromosome 14, the left end, if you will, that's higher in what nucleotides than average?
Higher in GC.
Yeah, higher in GC, right there. So it's got a lot more G's and C's than the chromosome average, if you just look at all of the G's, C's, A's, and T's in the entire chromosome. And the same thing is true on the other end of the chromosome. Near the telomere, the proportion of the nucleotides on the chromosome that are G's and C's are increased. Now, it turns out that the chromosomes don't have stripes just when you look at a chromosome. DNA by itself is not naturally pigmented, like I am not naturally pigmented. DNA has to be stained, chemically treated, in other words, for us to actually see it. Otherwise, DNA is clear. Has anybody done the DNA extraction from a strawberry sort of experiment? A couple of you have. You mash up strawberries, put on isopropyl rubbing alcohol, you take a hook, you spool all the DNA. What color is it when you do that?
White.
It's white. It's basically clear. The white is from salts. DNA doesn't have color. So to elicit these bands, to bring out stripes on the chromosomes, scientists chemically treat, and those are chemicals that specifically react to G's and C's or A's and T's. The reason that I'm showing you human chromosome 14 next to the banded version of human chromosome 14 is that what's a general pattern in the bands and the isochores? When you line them up, the bands, the dark bands, tend to be in regions of the chromosome where there are lots of G's and C's. And that's because the chemical that this chromosome was treated with reacts with guanines and cytosines and produces a dark pigment, and that's why those stripes show up on the chromosome in the pattern that they show up in. It's because every chromosome, when you move from one end of the chromosome to the other, has variation in the amount of G's and C's or A's and T's. And when you chemically treat the chromosomes, you can see those differences along the chromosome. And every chromosome has a different pattern. And that's why cytogeneticists do this chemical treatment. So when you look at chromosomes, you can actually figure out which chromosome is which. Everyone's got a fingerprint, in essence, a unique pattern of dark and white stripes. You can tell the chromosomes apart this way. Questions about that? That's a relationship between isochores and stripes, why chromosomes have stripes.
So generally the isochores will always be the ones that are striped, the darker ones?
So if I interpret your question, is it where the G's and C's are high that there are always stripes? Is it always a correlation between dark band and high GC content? Depends on the chemical you use to treat the chromosome. So in this case, this is called R banding, which is treatment with a particular chemical that you don't need to know, that's fine. There are other forms of banding that use different chemicals that bring out different patterns of stripes. So don't worry about it. But you should know that depending on the chemical you treat the chromosome with, you'll get a different pattern for the same chromosome. R stands for reverse banding. So this is the opposite pattern of what you would see if you did. So this is R banding. If you did C banding, for example, that is a type of banding that stains centromeres. Where's the centromere? I cannot. What shaped chromosome is this? Is this like an acrocentric or a metacentric? It's hard to see where the centromere is. Where do you think the centromere is? Is it in the middle? Let's pretend it's in the middle. Let's say the centromere is right there. If you did centromere banding, there would be a dark stripe in the centromere, but nowhere else. So there's a particular chemical that we can treat chromosomes with that reacts with the DNA that's at centromeres. The main form of banding, aside from R banding, is G, and G has the opposite pattern of R. So that's why R is called reverse banding. G was sort of the first type of banding that was done, and then somebody came along and realized that, oh, if I do treatment with this other chemical, I get exactly the opposite pattern of dark and light stripes. So G gives you one pattern, R gives you the reverse pattern. Where there's white in G banding, there's dark in R banding. In case you ever happen to become a human clinical cytogeneticist and spend all your time in a laboratory banding other people's chromosomes. Of course, the reason people do this, why do you think people do this sort of analysis, human chromosomes? This is a job. This is a career you can have. I know people that sit in laboratories all day and just stare at human chromosomes.
To figure out specific genes that maybe are helpful for human health, so they have specific spikes in dark areas that identify them.
Right, so you can learn something about genetic conditions in humans or other species by looking at a chromosome and knowing what its banding pattern should look like, and then saying, hey, wait a second, that's human chromosome 14, but that black band is missing. That tells the cytogeneticist, the clinician, that there might be a deletion of that part of the chromosome from that person that you're looking at the chromosomes of. They might be missing some genes. Their chromosome might have broken and lost that part of the chromosome. What else can we learn? What's a really common human condition that involves changes in chromosomes that causes a phenotype?
Trichromy 21.
Trisomy 21, Down syndrome. You have an extra copy of chromosome 21, 3 copies instead of 2. Those individuals develop Down syndrome. That's something you can tell by looking at people's chromosomes. In fact, that's how you tell. So you have to take cells from the individual, look at them on a microscope slide, and count. Say, oh, 1 extra copy of chromosome 21.
So is that how they match homologous pairs? They're matching the two bandings?
Exactly. So when you have an organism like humans, where we have 2 sets of every chromosome, you know which 2 chromosomes from that huge mess of chromosomes are the same chromosome by looking at the pattern, and we'll do an exercise regarding that in a little bit. So that was why chromosomes have stripes. Different banding patterns elicited by different chemicals we can treat chromosomes with. Okay, so here you see, for example, I've shown this before, a representative human set of chromosomes with banding. And you can, if you stare at it long enough, you can tell that each chromosome differs not only by where its centromere is, but how long it is, and then its pattern of bands. With those 3 pieces of information, clinicians, cytogeneticists, can line up the chromosomes like this. Pair the 2 copies of every chromosome each of us have together to create a karyogram. Now, I also realized that last class I didn't spend a lot of time talking about, and there's a purpose for this. We're going to talk more about this later when we talk about mitosis and meiosis. I didn't go into a lot of detail about how you determine the karyotype, 2n equals 46, based on what you see as chromosomes. So I wanted to give you a little bit of a primer on how you do this, especially related to that formula, 2n equals 46. So there's a question about this last class. How many different chromosomes, this is, by the way, from what sort of sex individual, male or female?
Male. It's got a Y chromosome. It's a
So
dude.
Y chromosome, so it's a dude. Y chromosome. So if, in a human, if you've got a Y chromosome, normally you're male. We'll talk about when that doesn't happen at a later class. So male. How many pairs of chromosomes do humans have? How many unique different chromosomes do we have?
23.
They're numbered up there, so hopefully it's pretty easy. There's 1 through 22. So when we talk in the future about chromosome 1, that's chromosome 1. It's our biggest chromosome. That's its unique pattern of bands. We know that's chromosome 1. Then it goes down to chromosome 22. The numbering occurs by order of size. You don't need to know that, just in case you want to impress your friends the next trivia night. So the biggest chromosome is numbered 1. The second biggest is chromosome 2, and so forth. So get down to 22, and then there's, if you're a female, 2 X chromosomes. If you're a male, an X and a Y. That's our 23rd pair. So each of us has 23 different chromosomes, 2 of them in each of our cells. So if there are 23 chromosomes up there and 2 of each, how many total objects do we see up there? whatever 2 times 23 is, right? We got 2 of 23. It's not a math class. So what is n?
23.
N is 23 in this case, which is the total number of unique chromosomes in that individual. Chromosome 1, chromosome 2, chromosome 3. So N is the number of different chromosomes that an organism has. And for a diploid, which are organisms that have 2 copies of every chromosome, that's where the 2 comes from. So 2 copies of every chromosome is the 2. N is the number of unique chromosomes, number of different chromosomes that organism has. So 2 times 23 gives us the 46 objects that we see up here. Now it's your turn. Any questions about that? Because it's time for some practice. So here's what's a drawing of chromosomes called?
Karyogram. That's a picture.
Drawing. A cartoon of the chromosomes like this is ideogram. So go for it. Work with a partner, work by yourself, share your answer. What's the karyotype of this set of chromosomes? Okay, I'll give you about a minute to do that. All right. So how do you approach solving this problem? What's the first thing you do?
Find the pairs.
Yeah, that's right. If you've been reading the advice from former students, it's make sure you read the question fully, read it again, make sure you understand. So what's the karyotype? Arrange them by size, or better than that, there's bands up here, so you can arrange them by size, and that's best here. So which one's the largest chromosome? You want to double check always. So there's 2 copies of chromosome 1. How do I know that those are identical? How do I know those are both chromosome 1, say, in this case? These are not meant to be human chromosomes, by the way. The bands match. So I've labeled both of these, what I'm calling chromosome 1, at the same end. So you should have the same pattern of bands when you move from one end to the other of both of those chromosomes. Okay. So what's the next biggest? This one's got an interesting, unique pattern of 3 bands close together at the end, so I'm going to call that chromosome 2. You don't have to number these the same way I did, but that's the first step. We also need to make sure that we're looking at the same centromere positions. That's another way to know. So how many chromosome pairs are there?
5 pairs.
Yeah. How many objects are shown up here? 10. So whatever our karyotype is going to equal 10. How many copies of every chromosome is there? In this case, there are 2 of every chromosome. So now it's just division. 2 times something equals 10. That gives us n. That something is n. So there's 5 unique chromosomes. So karyotype. 2n equals 10. Yeah?
Aren't there 2 chromosomes per pair, though?
Oh, don't have to. This is, no, I'm kidding. I'm glad you asked me that. This is a really, really, really common issue with the study of genetics, which we're not quite equipped to answer. So the short answer is yes. Chromatids versus chromosomes, mitosis and meiosis, which we're heading to shortly in class. But you're absolutely right. If you don't want to hear this yet, close your ears. Each one of these things is a chromosome. So really, how many copies of chromosome 2 are we looking at? There are really 4 here. But when humans look at chromosomes like this, we often are inclined to say that that thing is an object. We colloquially, that is in common language, call that thing I circled in red here, we call this a chromosome. It's really 2 lying next to each other. So we're going to talk more about the nomenclature necessary to deal with that, chromatids and chromosomes, in a chapter soon to arrive in class, but we're not quite there yet.
So is that a chromatid?
Those are 2 chromatids next to each other. This thing.
So it's not the chromosome?
That's 2 sister chromosomes. That's all I'm going to say about it at the moment.
It's a duplicate
Come talk to me after class, and I'll explain in person. So the thing I hope you take away from that is how to look at an image of chromosomes, an ideogram or a karyogram, and calculate the karyotype to arrive at the karyotype. Now, one of the things I asked you to do for class today was to spot a recognition sequence or a restriction endonuclease. And I've just thrown up in here, and you've got these on your PowerPoints or your PDFs as well, some answers from some of you. I've made them anonymous. I always will. If somebody who wrote this has a question or wants to claim responsibility for this, I'm always happy to let you make that decision in class if you want to. So I asked, find and underline a restriction endonuclease recognition sequence. That was one of our tasks for last class, which means it's going to be something that's important to know for an exam. So remember, all of you have the generic exam questions already. They're the tasks that are listed in every chapter of our class manual. Those are the things I'm going to be testing on. Trying to be really transparent about that. So this individual discovered a restriction enzyme site for the enzyme EcoR1, which recognizes that sequence GAATPC. You might have figured that out if you watched the video on restriction endonucleases, but for all of us, how would you find a restriction site? Those of you that did this assignment, how did you go about doing it? Help us out. What do you look for when you try to find a restriction endonuclease site? So I've got the word palindrome, and I've got a great hand gesture going on, and it's not involving middle finger. So we're looking for a palindrome, and I didn't get a chance to talk in detail last class about what we mean when we talk about a palindromic DNA sequence. Does everybody know palindrome? Something that reads the same forward and backwards? Like, what's a good example in English? Which? Race car, Hannah, mom, right? So let's stick with mom because I know how to write mom. So if you read it in one direction, it reads mom. If you turn the letters in the opposite direction, it still reads mom. A really famous one is a man, a plan, a canal, Panama, which reads the same backwards and forwards. That's not quite what we mean when we talk about a DNA palindrome. It's close. Because, of course, GAATTC, when read backwards, is what? Yeah, CTTAAG. So when we talk about DNA palindromes, we're not talking about reading one strand forward and backward. What we're talking about is what?
The opposite strand, reading the opposite way.
Remember, DNA is double-stranded. And a convention, if you take anything away from today's class, this might be it. Pay attention. Our convention as geneticists is just to write a single strand of DNA sequence when we communicate with each other about DNA sequences. Why?
Because you can determine what the opposite strand is based on that one line.
Right. If I give you one strand of information, that was an exercise I assigned a couple of classes ago, you should be able to figure out what the other strand sequence is. So what's the other strand of GAATTC, starting here? CTT, just base pairing. A's with T's, C's with G's. So there's a 5' end and a 3' end. That is our convention. We write one strand of DNA sequence, and we always start on the left side with the 5' end, and we end at the 3' end, just like was done down here. So a palindrome. Now do you see why that's a palindrome? Up here. When you read, which direction do you always read DNA sequences? Starting at the 5' end, going to the other 3' end. So read both of those in the same orientation, 5' to 3'. Top strand, what's the sequence? GAATTC. What about the bottom strand? What's its sequence? It's the same. GAATTC. It's a palindrome. It's a DNA palindrome. It reads the same backwards and forwards. You just have to think about the second strand in the case of a DNA palindrome. Lots of proteins that bind DNA, that interact with DNA, read the sequence. Proteins don't have eyes, they don't read. But proteins that physically interact with DNA, which we're going to be talking about a lot in this class, physically, like Braille almost, read the sequence of DNA by protein touches the DNA and can feel the structure of the nucleotides it's touching. And most proteins that interact with DNA read palindromes. And that's why I'm making a big point about this. Palindromic DNA sequences. Restriction endonucleases or restriction enzymes tend to be a large class of proteins that do this. So this is not the only answer. This is one answer. And I put some other answers from your fellow classmates in this set of slides. Some are right, some might not be quite right. Does anybody spot another palindrome up here?
GGCC.
Where's that?
Top line, right above this 3'.
Geez, so let's switch colors. GGCC. So if I write both strands, 5' to 3', GGCC 3'. So it's another palindrome. There are lots of palindromes hidden in this sequence. Why did I do this to you? Pardon? Recognize patterns.
I have a question.
Please, no, that's fine. Love questions.
So when you're looking for palindromes, do they have to fall on the 3 codon?
No codons yet. Don't have to worry about codons yet. We're just looking for DNA sequences that, so it can be, so what's the easiest type of palindrome to spot? Pardon? GGCC, well, there's a smaller palindrome inside that,
GC.
GC. What about just a single letter? A. We could get in a whole conversation about whether a single letter word is palindromic. I is a palindrome. Well, actually, both types of I, letter I and EYE. But, right, so if I ask you, remember, the reason that I gave you this assignment is not only to make a point about genetics, which is an important point, but to remind you two more important things about the process of science. One is there's not always one right answer. In this case, there were multiple right answers. This thing is riddled with palindromes. And why make it hard on yourself? Go for the easiest possible answer if you want to. Look for a simple palindrome. You don't have to find one that is actually a restriction site, which in this case is GAATTC, is a sequence that a specific protein called EcoR1 recognizes. Last question, and pretty quickly, how would you figure that out? If you spotted a palindrome, like GGCC, for example, how would you figure out, right now, if I said, your grade is on the line, what restriction enzyme does GGCC interact with? What would you do? Or your favorite search engine. It doesn't have to be Google. I'm not paid by or endorsed by Google, but yeah, Google it. So somebody please, Google GGCC. I have no idea what is going to happen. I've never Googled GGCC before in my life.
Hemophilus aegyptus.
Discovered since 1970. It was the third endonuclease to be isolated from
Hemophilus aegyptus.
bacteria, and has a weight of 337. I don't know, that doesn't give me actually.
Okay.
a
So,
quantifier for that.
yeah. So I heard an enzyme called Hey3. Hey. I don't know. It's from, and this is, again, an important point. Thank you for pointing this out. The way that enzymes are named is the first letter of the genus name of the bacterium. So Haemophilus is the genus name, so that's where the H comes from. The AE is from Egyptus, AE from Egypt, but starting with an A. I don't know who spells that way, but. And then this is the third, as you read, thank you very much for doing that, the third enzyme that was isolated, so they use Roman numerals. So this is Haemophilus aegyptus, the third restriction enzyme. That's how you find out these things, if you want. Google it. You don't need to memorize restriction enzyme names or the sequences they recognize. I just want you to be able to find this information if you need it. Let's pass some other ones that I dropped in here for fun. But if you have any questions about your answer, please let me know. So that's all I was looking for. So can you spot a palindrome? And then afterwards, if you want to, can you answer the second part of the question? Can you figure out what enzyme recognizes that palindrome? The other thing that I asked you to do for today was to try finding the DNA sequence of a gene. I gave you the name of the gene. You needed to figure it out. Most of you that submitted this went to the NCBI website. There's a lecture video on using NCBI. It's a pretty short one. That was successful for most of you that turned in this assignment. You found some sort of DNA sequence related to the human, Homo sapiens, BRCA1 gene.
I have a question.
Absolutely, please.
There's like 3 that popped up, and I'm wondering, is there a specific one we're supposed to use?
Multiple possible answers.
Okay.
Yep. And that was one of the things that I wanted you to discover about the NCBI database. All that is, for those of you that aren't aficionados, which none of us are, NCBI, the National Center for Biotechnology Information, houses the US-based repository of all the known DNA sequences in the world, basically. trillions and quadrillions, literally, of letters of DNA sequence. And when scientists upload these DNA sequences, when they take their favorite organism and extract some DNA and determine the sequence of it, they submit that information to this publicly available database, and they add some useful information like the species it's from, the name of the gene, and other things. And that's all what's found up here in that FASTA name, right? So this is from Homo sapiens humans. It's the BRCA1 gene. The problem with this particular submission is what happens after Homo sapiens BRCA1 gene, which we haven't talked about yet. But again, the point was for you to discover that in NCBI, there are tons, almost literally, of sequences for any particular organism and any particular gene you're looking for. So there'll be loads of records. Some of you probably found 10,000 or more different records in this database for BRCA1. I'm just hoping you pick one. So this person, I asked, how many times does this restriction site, GAATTC, appear in the sequence? Said it doesn't. There's one that's close. That's fine. That's correct. That DNA sequence that's 6 letters, GAATTC, doesn't show up here. That's okay. Bonus if you calculate how often that sequence should appear in the gene. This is correct, absolutely. And this has something to do with that Socratic quiz that we just went through. Okay. This is an important one to talk about. Full circle. Isochores, banding, restriction enzymes. Talk with a friend or a non-friend colleague that you happen to be sitting next to. Frenemy. Discuss this for 1 minute. We'll come back together. We'll discuss the answer. Where are you more likely to find a restriction site for that enzyme? Okay. Go ahead. We'll come back together in 1 minute. All right, noise is winding down, which means everybody's got an answer. So, volunteer?
On the end.
Should I call on somebody? What? Why the ends? And by the way, I'm not going to let you answer every question in class. I appreciate your involvement. Go for it. Why the ends?
There's a higher percentage of GC at the ends than average, so you would expect that's what they are.
So if we're looking for 8 nucleotides that are all G's and C's, it's not going to be as likely where? In the middle of the chromosome where there's a lot more A's and T's. Here's 8 nucleotides that have 0 A's and T's in that sequence. So where are you most likely to find that sequence on this chromosome? If you could pick one specific spot on the chromosome. Tell me when to stop. Hey, I am actually looking right at that peak. Right? There's the spot on the chromosome that has the highest number of G's and C's in one spot. You're most likely to find that specific 8 nucleotide sequence there. That doesn't mean it's going to be there. That doesn't mean it's not going to be elsewhere. It's just that's the most likely place you're going to find a bunch of G's and C's.
Dr. Ross?
Yeah.
So I was looking at some videos, and some of the shorter restriction endonucleases, like the Haemophilus aegyptus, right? It seems like there's not that many, there's only 4 base pairs.
Right.
So it seems like, over the whole course of the chromosome, you probably have a lot of repetitions of that just based on probability.
Yes.
And the video that I was watching was explaining that there's proteins involved in selecting, when and where these endonucleases are used. So I'm just asking, what kind of oversight do these proteins play, and how is that, how are they so picky that they know exactly where to cut on a chromosome strand?
I'm going to defer. I love the question. I want to defer to a later class where we're going to talk specifically about how restriction enzymes recognize sites. I'm happy to tell you in private.
I was just wondering, because the Haemophilus aegyptus...
Because I don't want to distract everybody else.
Yeah, it was a disease. So I was wondering if the length of the restriction site affects the frequency of cutting.
Yes. So with that, so again, to summarize, the length of the restriction site, the sequence that the protein recognizes before it cuts the DNA, which is what restriction enzymes do. They find that sequence, and then they actually physically break the 2 strands of DNA where they bind. That's the part that's called restriction. The endonuclease is endo breaking, is a nuclease. It breaks the DNA. Endo means inside, so it's an internal break of the DNA. And the restriction part has to do with bacterial defenses against foreign invaders. So bacteria have these enzymes because they use it to restrict viruses and viral genomes DNA from the bacteria. So these enzymes are part of bacteria defense systems that recognize foreign DNA and cut it up, try to destroy it. And absolutely correct, the longer the sequence, the less frequently it will occur in the genome, and that has something absolutely directly to do with 1 or 2 of those Socratic questions. So I'd love to go straight to that, actually. Let's wander quickly through our Socratic quiz answers to see if there's anything that needs discussion. Let's see. What organisms have a larger genome than humans? Plants, yes. Some amphibians. No on the bacteria. No bacteria have genomes larger than ours. So A and B are correct. That was from one of our videos. Consensus sequence. Yeah, so let's talk about consensus sequence.
Has to be over 50%, right?
Depends on who you talk to. So how do you build a consensus sequence? You've got 4 different sequences here, shown right underneath the number 4. I'll circle them in red. So let's name these. Oh, I shouldn't use letters, should I? Let's use Roman numerals since that's fun. 1, 2, 3, and 4. Wow, yeah, those are tiny I's. If I zoom in, you'll see the full lowercase i's there. 1, 2, 3, and 4, top to bottom. Everybody okay with that? So we've got 4 different DNA sequences. They're all lined up next to each other. What does it mean? What's the definition of a consensus sequence?
To look at each column.
So you look at every column, and you ask yourself what? So there's the first column. What's the most common? You're looking basically for the average. Consensus, average. We could get a consensus feeling for how everybody feels about my role as an instructor in this class, right? That would be getting the overall feel, the big picture. So that's what we're doing here. Column 1, what's the average nucleotide? What's most frequent or most common? A. Okay, last column, A. Second to last column, it's 100% A, so we don't even have to use brain cells. So then the problem comes in the second column, which now I'm drawing all over the place, but there are 2 G's and there are 2 C's. So what's the rule? Is it 50% or higher? Right here we've got 2 sequences that have G and 2 that have C. So it's 50-50, G or C.
Did you just pick both?
I'm happy with your logical answer, whatever it is. So we've got one vote for pick both. In that case, the correct answer is there are two different consensus sequences. They only differ there. One has G, one has C. So you could say it's a tie. We don't have an average. We have two averages. Another way to write that would be, which is genetic nomenclature, G/C in parentheses for that second character, if you were going to write out the consensus sequence, and then TCGAA. So reasonable people might say there is no consensus because there's a 50-50 half 2 and 2 at that second column. Or, so you can say both. You could say there is no consensus, as long as you justify your answer to me and you show that you understand the concept of a consensus sequence. So I was thinking A and D are the correct answers, but you might be able to convince me otherwise if you have an argument for that. Why do we care about consensus sequences? You might get to that. Any thoughts?
Is it important in evolutionary biology? Or?
It's important in evolutionary biology. It's important in the study of us and our differences. Because every one of us is a little bit genetically different. So depending on those 4 sequences, might be the sequence of the same part of your genome and my genome, and your chromosome, and your chromosome, and your chromosome. Because we all differ a little bit in DNA sequence. But it's kind of hard to discuss our individual sequence differences. Sometimes it's more useful for scientists to say, this is the human genome sequence, which we're going to talk about in a couple minutes. The point is, there is no the human genome sequence. Why? There are individual variations, and how many individuals are there on the planet?
8 billion.
Some number of billion. Right? So there is no one human genome sequence. By the way, it was published in 2001. So we've known about the sequence of the human genome, in quotes, for over 15 years. Except there isn't one person's genome that stands for all of our DNA sequences and our differences. So that's why consensus is important. It gives scientists sort of a baseline. We say on average the human genome sequence is this. That requires us to be able to calculate or to compute the consensus sequence, because we all have genetic differences, thousands of them. Okay. Most of you from this Socrative quiz calculated this. Not all of you. I'd say maybe a close majority. So, questions? If you look at any part of the human genome or any sort of DNA sequence, how often will you see an A? How many letters are there in the DNA alphabet?
4.
Sorry, I was holding onto my stylus. Needed my thumb. 4, right? A's, T's, G's, and C's. So on average, every 4 nucleotides in a random DNA sequence, you should spot an A. So once in every 4 nucleotides. Because for every position in a DNA sequence, what's your probability that there's an A there? 1 in 4. Okay, now, a lot of you got that. The next question was something to the effect of what happens when you're looking for 2 A's in a row. So let's do that. That might not be the question I asked. What's the probability of spotting an A next to an A? What's the probability at this second spot? What's the likelihood? you close your eyes and you just look at any spot in the DNA and you see an A. It's still a 1 in 4 chance, right? That probability hasn't changed. So the only question is, starting to get that deer in the headlights, holy crap, we're talking about probability and statistics now. It's easy. This is the only thing you'll have to remember about statistics and probability for a while. It's what you do with those two probabilities to calculate the likelihood that you see those two A's next to each other. AA. You add them together, you multiply them together, you square them. What do you do?
A and A, it's multiple.
You multiply. So the probability of seeing two A's next to each other is, yes, Siri? 1 in 16. Hey, Siri, what's the probability of finding two G's next to each other? That's true. This is the weird thing to me, and that's just because I'm sort of a statistics and probability newbie. It doesn't matter which two nucleotides you put down there. It doesn't matter if it's AT, GG, GC, TT. Any two nucleotide sequence, every 16 nucleotides you look at, you should see that pair once. So what's the probability of seeing any 3 nucleotide sequence? Any 3 nucleotides. I can make it up if we want to make it concrete. What about AGA? I don't do probability, I'll do frequency. 1 in how many base pairs? Every 1 in, a 1 in 4 chance of having an A, a 1 in 4 chance of having a G next to it, a 1 in 4 chance of having an A next to it. So the probability is 1 in 64. So if we have a 4 nucleotide sequence recognition of a restriction enzyme, it's looking for GGCC, for example. So restriction enzyme, what was it? Hey3? Hey3. Oh, sorry. How frequently?
1 out of 256.
1 over, yeah, it's a quarter times a quarter times a quarter times a quarter. So 1 in every 256 nucleotides, you should spot CCGG or GGCC or any 4 nucleotide sequence. So the longer the recognition sequence of the restriction enzyme, the less frequently, by chance, it shows up in the genome. Questions? Saw a hand up. Back.
So if you, the Isochore, like if you have a DNA sequence that is predominantly, ET or CG, what correlation does that have with this?
Okay, so my question was about composition of the sequence you're looking for. And we're talking about frequency here. This is in a random DNA sequence. How likely are you to see any particular sequence? That's the calculation. You take it to the bank. But except for this whole situation about isochores, for example. So yes, in a random DNA sequence, this is true. But we just saw in isochores that the DNA composition of a chromosome is not even from end to end of the chromosome. Starts out with high GC content and then drops to low in the middle. So when I ask questions about this, I'm expecting you to assume random DNA sequence. But you're absolutely right. In real life, which is what I always try to talk about in genetics because it's a very applied field in biology, yeah, you would have to think about, if I ask you about distribution of nucleotide sequences. High AT is more likely in the middle of the chromosome, high GC on the arms.
Is that a universal trait
Among species? No.
in all life?
Yeah, right, correct. That's what I meant. Among species or in different species, that pattern does not necessarily hold up. Of high GC at the telomeres and low AT at the
Only in
centromeres.
certain species.
Some species. Yep. Thanks. Always good to challenge the model, because the model, or what I tell you, is not always true. And if I know I'm lying, I will tell you I'm lying. But I always appreciate having those challenges.
I remember the anoles were kind of random.
Right. So that of the case study paper we read talking about isochores, that species didn't have variation really from end to end of their chromosomes. Okay. Did anybody get that? You can paste the accession number into NCBI, which is what I hope people did, because you're always free to use the tools you have at your disposal. EGF receptor, I think, epidermal growth factor receptor. I don't remember which species. I think I have that somewhere. Oh, Drosophila, fruit fly. Okay, so I'm going to tell you a brief story, then we're going to get out of here and enjoy the 100 and whatever odd degree weather is going on outside. Aren't you thankful for air conditioning? And the library, probably. Okay, quick definition of a genome. Easy. It's all of the DNA that you have in a cell. Now notice I said cell. What could I have said instead? What was that? Nucleus. Again, reasonable scientists disagree.
Is mitochondrial DNA really part of your genome, though?
Yes, that's my opinion.
You think it is?
I do. So mitochondria, everybody, okay, I know most of you have taken biology 1A or 1B, so you should, and you've taken high school biology and so forth. So I'm going to go out on a limb here. I'm going to hope you're going to hang with me and help me out, okay? I'm going to ask you to say this with me. I'm not going to tell you what we're going to say. Mitochondria, this should sound familiar. Mitochondria are the blank. So I'm going to start by saying mitochondria are the, and then you're going to help me out, okay? Mitochondria are the powerhouses of the cell. Okay, good. So at least some of you have heard that before. Mitochondria have their own DNA. Right? Mitochondria and chloroplasts, organelles, also have DNA, not just our nuclei. So yes, mitochondria have their own genomes. Depending on who you ask, I believe that that's part of the human genome. Different scientists don't agree with me.
What?
Some of us do, some of us don't. Well, it's all a matter of definitions. So in general, when people talk about the genome, they're silently referring to the nuclear genome. That is all of our 23 pairs of chromosomes if you're talking about a human. I add in the mitochondrial genome as well. And again, I don't particularly care which definition you use with mitochondria or without, if you're talking about an animal, or with chloroplasts or not, if you're talking about a plant, as long as you can explain to me that that's the definition you're using. So either one's fine. I don't mind. So it was about, goodness, back in the ether of time, I suppose, that Mary Claire King published this really, really, really controversial study comparing our closest relatives, chimpanzees, our closest related species, chimpanzees, and us, or at least, we look pretty similar back in the time when Neanderthals roamed the earth. I mean, they look pretty similar, right? There are no Neanderthals around these days. So instead of having Neanderthals, what King and Wilson did was they compared, they didn't have DNA sequencing technology at the time, or at least not enough, so they compared protein sequences of chimpanzees and humans. So they took some proteins out of chimpanzees, they took the same proteins out of humans, because they wanted to answer the question, how similar at the molecular level are chimpanzees and humans? Nobody knew at the time. This is groundbreaking, and of course, anti-evolutionary people had a fit, which is fine. That's their right to do so. So that's what King and Wilson did. They took proteins out of chimpanzees, proteins out of humans. They looked at the sequences not of the DNA, but of the proteins, the amino acids that made them up. And what they discovered was, so this is a list from their paper. This is a list of the different proteins that they took out of chimpanzees and humans. This is how many amino acid differences there were between the chimpanzee version of the protein and the human version of the protein. And this is, in the third column, amino acid sites basically means the length of the protein in amino acids. So what do you notice? Humans and chimpanzees, pretty similar. Right? Depending on which protein you look at, there are no differences in protein sequence, or there are just a few. So overall, they looked at 2,633 different amino acid sites in these proteins, and there were only approximately 19 differences. So what's the percent difference between a chimpanzee and a human? Just looking at these proteins. It's, you divide those two. Yeah, so it's, what they calculated was there are 8.2 differences per 1,000 amino acids. Or we are 99.2% identical in protein sequence to chimpanzees. And that's where, from then on, conversation in the country and around the world got really interesting about whether or not we're related to chimpanzees or not, and how closely related, and most interestingly, to me at least, and I hope to convince you of the same, how such a small difference, 0.8% in protein sequence, slightly different in DNA sequence, how such a small difference makes such a big difference in how chimpanzees and humans look and act and behave. So we're very similar at the DNA sequence level to chimpanzees. Of course, then we ask the question, how different are two humans? If humans and chimpanzees are 0.8% different, how different are you and I? Do you want to know the answer?
Yeah.
Do you really want to know? Are we as different from each other as chimpanzees are from both of us? Am I just a fancy chimpanzee who teaches a genetics class? Basically.
I read somewhere that we have like 91% similar DNA to a banana.
So we're something like humans, human to human, Homo sapiens to Homo sapiens, are something like 99.9% identical. That is, if you look at if you look at 1,000 base pairs of your chromosome and mine, 1,000 base pairs, there would be one difference. Which is
that DNA and protein?
That's DNA.
Okay.
And that's even more interesting because then you start, look around. Seriously. Look around at all the diversity we have in this room. Male, female, tall, short, etc. That's all due potentially to that 1 in 1,000 differences in nucleotide sequence between us. So very briefly, because I know we're just a couple minutes out, that was the case study for today. It was a recent study where, for the first time, remember I mentioned a while ago, there is no one human genome sequence. There's a consensus. This was the first time, this publication that I hope some of you looked at least, this is the first time that an individual human's DNA sequence had been published. So we knew it wasn't a consensus. We knew that was that person's DNA sequence. Right? No averaging. And the reason this is controversial, which we'll talk about in future classes, is because would you want somebody else to know your genome sequence? There are pros and cons. So we'll talk about that and its impact on society. So who was the donor, male or female? Male. So hopefully you remember from earlier in this class, today, Y chromosome, male. So I just mentioned to you how this genome sequence is different from the previous ones, and that was in the manuscript if you happen to catch that part. Earlier publications that had the human genome sequence in them were averages of multiple humans. In this case, I think they said it was 5. And this is the single human genome. Right? Yeah, there. Based on shotgun reads 5 individuals, previous version. This time just 1 human. The most interesting thing I thought, and you don't have to agree with me, hang on, just one more data slide, and then I want to wrap up, tell you what we're doing for next class. You get one person's DNA sequence. Each of you have 2 copies, I think. I'm going to make a guess. Each of you have 2 copies of 23 chromosomes. I could be wrong. Where did you get each of those copies, one set N, 23 of your chromosomes? Your dad. The other 23 came from your mom. So by having his genome sequence, this is Craig Venter, the individual whose genome was sequenced, now we have the DNA sequence of his parents. So this table is telling us how different his parents are in DNA sequence, basically. By taking his 2 copies of his chromosomes and sequencing both, they get, that's what this number of variants means. Differences, variants. Differences between his 2 sets of chromosomes. In total, there were 12 million base pairs worth of sequence difference between his parents. That is, within his own genome, his 2 sets of chromosomes, there were over 12 million base pairs worth of differences. So what is that in percent? How big is the human genome? This is just like the human versus chimpanzee calculation. So I'll leave you to do that, because I want to get to the last couple of non-topical slides. So it's 12 million out of 3 billion. So his parents are genetically different. Of course, they're still humans. And he still had a son who grew up to have his genome sequenced and published. So again, these are, now you've got a record of a few of the vocabulary items that are important for class. The tasks I hope you're able to do. We'll talk about numbered DNA sequence alignments a little bit more next class. And finally, so we've got what to read specifically in the case study for next class. Focus on figures 4 and 6 and answering that one question. Come to class able to discuss, hopefully. And then I'll be having another, I think this is almost the last survey to put online. It's worth a point. These surveys don't really have due dates. Try to do it by the next class period, please. I appreciate the feedback. So for next class, nothing to do that's worth any points other than that survey. Okay. Watch the lecture videos, read the textbooks, come to class.